After explaining how Drosophila wing develops, I'd like to introduce a model that is used to simulate this process. Developmental mechanisms, cell behaviors, etc. can be represented by mathematical equations, so we can build models to simulate organ development. The model that I'm going to show is the same that was used by Ray et al. In the 2015 paper. The idea is to incorporate everything that is known, all the developmental processes that are known that happen during development, and try to reproduce the whole process in silico. So this model is based on a formalism that is called vertex model, where each cell is represented as a polygon consisting of a set of vertices in blue in the figure, and each of these vertices is at the intersection between 3 different cells and 3 different edges. Vertices are moved according to some equations of motion in a way that their movement tries to minimize the overall the free energy of the system. The simulation procedure is as follows. One vertex is selected at random and it is moved to a random location. Energy is calculated in the previous location and also in the new location, and if the free energy in the new location is lower than in the previous location, then the vertex is moved permanently or to the new location. However, if the free energy of the new location is higher than in the previous location, the vertex returns to the previous location. With a low probability, it can stay in the new location, which accounts for noise in the simulation. This equation here is the one that is used to calculate the energy, and it has 3 different terms. In the first one, the summation is over the different cells that are in touch with a vertex. The second term is over the edges that are in touch with a vertex, and the third term is also over the different cells that are in touch with a vertex. So let's see each of these terms in more detail. In the first term, A alpha represents the current area of a cell, and A0 represents the equilibrium area of a cell. So the equilibrium area is the area towards which a cell tends. It's like the favorite area of a cell or its preferred area. When current area and preferred area are very different, this term gets larger because this radio will be bigger than 1 or smaller than 1, so the overall absolute value of this term will be higher, so energy will be higher, and it will be more unlikely that a vertex moves to a position where this term is higher, where this quotient is very different from 0. However, if the current area and the equilibrium area are identical, this term will be 0, so the energy will be lower. Okay, it will have a lower value, so it will be more likely that a vertex moves to a direction where this ratio is equal to 1. In the second term, this is a parameter that represents edge tension, and this L is the length of an edge. So if when tension is positive, the term becomes larger the longer the edge. It's like if you pull a string, if a string has a lot of tension, the more you pull it, the bigger the force it makes to shrink again. So long edges, edges that have a positive tension will increase, will have a bigger influence in the energy function the larger they are, and they will try to shrink. However, tension can also be negative, so edges try to become larger. The third term tries to make cells rounder. It tries to simplify their shape. This is a parameter which is perimeter contractility, and this is this L is the perimeter of a cell, which is divided by the area. So if the perimeter is large compared to the area, the term will be larger. So vertex movements will try to avoid that. Will try to avoid the perimeter is large compared to area. So shapes that have a large perimeter compared to area are, for instance, a star or a shape that is too long, and shapes that have a lower perimeter to area, right, are a circle or a regular hexagon. So cells will try to, or will tend to have these more rounded or regular shapes. The model also includes changes in cell topology. For instance, T1 transitions, which I already explained in another video, which is simply a change in neighbors. Like here, horizontal, the left and the right cell stop being in contact, and the upper and lower cell come to be in contact. Then T2 transitions, or cell extrusion, where one cell gets smaller and smaller until it is extruded from the tissue. And finally, cell division, which can be oriented according to Hurwitz's law or other factors. So I'm going to show you a few simulations of the system. This is a very long simulation. It's about, well, it's 500 million moves, but as you can see, nothing happens because the tissue is almost in equilibrium. So the only thing that happens is that the border gets more rounded because tension in the border is slightly higher than in the inside, but nothing else happens because there's no disequilibrium in forces. Everything is more or less close to, the energy is already minimized, more or less. In the next video, there's a lot of shape changes. So in the border of the tissue, there are some springs that are attached to the borders and pull from these springs pull from the tissue, and at the same time, cells have a very small equilibrium area compared to their current area. So cells try to get smaller, okay? And also there are lots of T1 transitions. Okay, so as you see, the tissue starts with a width of about 10 cells from up to down, and then it shrinks. First, the influence of the smaller area is bigger, but then these springs keep pulling from the tissue, so it gets more elongated, like a chewing gum that you pull from the sides. So this width of 10 cells becomes now the tissue has like 3 cells in width, and after a while it has 2 cells in width, and in some parts it starts having only 1 cell in width, like here, and then here it's only 1 cell, okay? Well, you can add more layers to the model. So you can implement gene regulatory networks, and you can make that some genes are regulating mechanical properties. So in this simulation, we have a morphogene which is secreted from a central band at a constant rate, and this morphogene causes an increase in cell area. Then cells with an area that is bigger to some threshold divide, and the cells that do not receive the morphogen reduce their area and eventually die. So this morphogen is like a trophic factor that is needed for cells to survive. Here the intensity is related to the concentration of the morphogen, and as you can see, cells in the center that receive more morphogen keep growing and dividing, whereas cells in the periphery get smaller and eventually die. And now this is a version of the same, but it's quite bizarre. So this is just to show some of the capabilities of the model. Here there are different cell types. One is the green cell type and the other is the blue cell type. The blue cell type is exactly the same as before, and now the green cell type responds differently to the morphogen. So the morphogen is secreted from both the gray and the black sides, and both cell types, the green and the blue one, grow in response to the morphogen, but the blue cells orient their divisions perpendicular to the gradient of the morphogen, whereas the green cells orient their division in the same direction of the gradient. So this is why they have this very strange shape. Okay, so this, of course, as you can see, they orient their division in a vertical way, which is the same direction of the morphogen, and the blue cells do as before. Okay, so how we do use this model to model the actual wing? Well, step by step, it would be very difficult to make a model that simulates all the whole process from the imaginal disk to the hinge contraction all at once. So first I'm going to show a simulation of hinge contraction. Here there are different cell types. The hinge cells have an equilibrium area that is smaller than their initial area, so they shrink, they contract. Then veins and vein borders have a higher tension, and the border is attached to some of these springs, like the ones that I showed before, that are analogous to the dampy protein. Here you see how there's one cycle of cell replication, and that cell replication also starts in the most proximal part of the blade, just like it happens in real wings, where division starts here and continues in the most distal part. You see how the hinge contracts, and this pulls from the blade and changes its shape. Okay, because the shape of the blade starts being more or less symmetrical, and quickly it becomes asymmetrical in the anterior-posterior direction. These different colors in the springs mean that they have different tensions, and this mimics different dampy concentrations in real wing, which happens. This is why you can pull from these springs and they get larger, whereas the others, since they have a higher tension, they are not stretched. So if you remove some of these springs, you can mimic the phenotype of a dampy mutant. Here you can see how the border is getting into the Next, we can simulate the process of convergent extension, which takes place between 4 and 6 hours after pupae. Here, cell fates are specified, so you will see different colors, but the mechanical properties are the same in all the cells because cells are not differentiated yet. So tension here is higher in the edges that go in the anterior-posterior direction, and as you can remember, in real wings, myosin II is planarly polarized, so its concentration is higher in these anterior-posterior edges. So this is what makes cells to elongate. Also, the equilibrium area is more or less like the current area, so cells do not change their area, they only change their shape. As you can see, there are lots of T1 transition that contribute to relaxation of cell shape and to shrinkage of the wing and elongation. And finally, we can simulate the process of extension, of expansion. This would be just an isometric expansion. So the cells increase in area, but they don't change their shape. The shape changes a little bit, but not too much. So it becomes a little bit wider. And well, that's all.